Hello everyone. In this lecture, I'm going to review the neuropathology and cytology of Huntington's disease, or simply HD. I'll begin with some of the more macroscopic alterations and then move down to the cellular and then the ultrastructural changes that occur in HD. If you have any questions, please comment below, and thanks for watching. So Huntington's disease is, in a nutshell, it's a devastating motor neuron disease that manifests primarily as a pronounced chorea or a involuntary hyperkinetic movements. Chorea is Latin for dance. And these hyperkinetic movements are thought to be a result of the loss of various subcortical brain structures. The overarching name for all these structures that you see here is the basal ganglia. So right here, this would be the basal ganglia. And basal ganglia is kind of a general term for all these basal ganglias. So the basal meaning below and ganglia meaning a subcortical gray matter region. And among these basal ganglias, the caudate and putamen are the most affected in HD. In these images, the caudate nucleus is just lateral to the ventricles, and the putamen is putamen, and it's just across this really thin white matter tract called the internal capsule. So we have the internal capsule running between the caudate and the putamen. Although the caudate putamen is the primary brain region affected in HD, there is nevertheless widespread neurodegeneration as the disease progresses, eventually affecting all brain structures. You see in grade 4, there's widespread degeneration. There's pronounced degeneration of white matter tracts. So the white matter, the axons running between the neocortex and the basal ganglia. And there's also enlargement of the ventricles as well as the brain shrinks. But either way, the earliest stages of the disease and the devastating symptoms of the disease are primarily derived from the loss of neurons in the caudate and the putamen. You can see in late stage diseases the internal capsule is still running through. You can see it right here, but the caudate is gone. Putamen is pretty much gone as well. The neocortex is vastly shrunken, and we're going to see a couple zoom ups here in a second. So the most affected region of the striatum, of the basal ganglia, is the striatum. And this is a close-up of the striatum. The primary structures are the caudate nucleus, right here, CN, and putamen, PT. Panel A over here represents an anterior section, and B, C, and D are kind of moving posterior, so they're moving backwards kind of. Areas in blue right here, the part of the putamen and a little bit of the caudate are functionally involved in sensory motor areas. Areas in yellow are associative areas, and areas in red are limbic. And the caudate nucleus is the first brain region to begin degenerating in Huntington's disease, the extent of which is also most correlated to CAG repeat length. So not only is the caudate nucleus the first region to show atrophy, but it's also highly correlated to the patient's CAG length in their Huntington gene. Functionally, the caudate nucleus is involved in sensory motor functions, where it basically receives potential movement operations from the motor cortex, and it filters out unnecessary movements, which is why on a macroscopic scale, the loss of this brain region results in sporadic, involuntary, hyperkinetic movements, chorea, because the caudate nucleus is unable to filter out in unnecessary movements that are arriving from the motor cortex. The caudate nucleus does this by harboring medium spiny neurons. Medium spiny neurons are GABAergic inhibitory neurons that control the excitatory motor inputs. And interestingly, these medium spiny neurons, which constitute 95% of striatal cells, are depleted by up to 40% in grade 0 individuals. So almost half of all striatal cells are gone even before a patient begins showing symptoms. The putamen, the PT, is also composed of medium spiny neurons, and it's the next brain region to begin showing atrophy. The putamen is involved in motor command inhibition, just as the caudate nucleus is, but it also has important roles in implicit learning. The last major structure in the striatum is the accumbens nucleus, the AC accumbens nucleus, and it's mostly involved in limbic and emotional control. And it's thought that the degeneration of the accumbens nucleus kind of leads to the more depressive symptoms that accompany HD. In this nice crescent violet stain, we can clearly see the degeneration of the striatum. So on the left, we have a control brain. This is a normal age-matched control. And on the right is a late stage grade 4 HD individual. We notice that the ventricle is huge. We have a huge ventricle. The caudate and the putamen are basically gone. It's not there anymore. So this striatum in this grade 4 individual is missing 95% of their medium spiny neurons, and the only remaining neurons at this point are mostly cholinergic interneurons that are simply passing through the area. Most of the remaining volume of the striatum at this point results from gliosis, or the proliferation of astrocytes and microglia, as well as just fluid-filled vacuoles. Basically, there's not much actually left in here other than passing cholinergic neurons and this internal capsule, which is a white matter tract. The internal capsule is a white matter tract that carries motor commands directly to the lower motor neurons, and it's somewhat intact. It's relatively spared in HD. We also see the claustrum and the accumbens over here. The accumbens is down here. So we also have the corpus callosum. Corpus callosum looks almost even larger in HD. So the idea here is that the toxic Huntington mutation seems to preferentially target and destroy medium spiny neurons of the putamen and caudate nucleus. And although surrounding structures do degenerate later in disease, the primary event is thought to be the caudate and putamen degeneration. So looking at the progression of disease across grade 0, so grade 0 to grade 4, pathologists find that the tail of the caudate nucleus is the first structure to actually show atrophy, followed by widespread atrophy of the caudate nucleus, caudate body, and dorsal putamen. Dorsal putamen is also affected early in disease. Nuclear inclusion bodies are present basically from grade 0, the prodromal stage of disease, right to the end. And we're going to talk about that in a little bit. In this slide, we're going to examine some of the circuitry of the striatum. So the primary input into the striatum is the cerebral cortex, mostly from pyramidal neurons from just about each neocortical layer, which we're going to discuss next. So these corticostriatal afferents are glutamatergic, meaning they're excitatory projections that activate medium spiny neurons. The cortex activates medium spiny neurons, causing these GABAergic neurons to deactivate downstream nuclei, including the substantia nigra and the subthalamic nuclei. This is substantia nigra and the subthalamic nuclei. So the striatum, activated by motor cortex deactivates subthalamic nuclei and substantia nigra. This renders these nuclei unable to inhibit the thalamus. And this results in increased stimuli entering and activating the cerebral cortex. So it's a confusing pathway, and I don't necessarily want to focus on the circuitry of the striatum, because there's a lot of double negatives and even triple negatives that we have to try and wrap our brains around. But the basic idea is that the loss of the striatum ultimately results in a hyperactive thalamus. And this hyperactive thalamus feeds excitatory inputs back into the motor cortex. Another way to look at it is that the subthalamic and substantia nigra nuclei, which innervate and tame the thalamus, are deregulated, causing the thalamus to excessively excite the motor cortex. I personally think of the striatum as a dam. It's a dam, and the motor cortex is the lake behind the dam. So the striatum, the dam, selectively permits water or motor commands to flow downstream to the muscles. When the dam is lost, when the striatum is degenerated, there's a flood of motor commands that result, and this leads to the hyperkinetic movements that we see in Huntington's disease. So the primary input into the degenerating medium spiny neurons are, again, these neocortical pyramidal neurons. Neocortical pyramidal neurons are the primary input into the striatum. And given their close association with the scene of the crime, they have also been studied extensively. So on the left, we have a control subject, and on the right is a representative section from an HD patient. Atrophy of the frontal, temporal, and parietal cortices has actually become a main neuropathological feature of HD. Layers 3, 5, and 6 of the neocortex appears to be the most affected regions, where postmortem studies show about roughly 30% loss of neurons, and this is compared to 88% loss of striatal medium spiny neurons at the same time point. So the cortex is definitely not as pronounced, but it's still a fairly large number of neurons have been lost, so it's still very relevant. Some lines of research actually suggest that cortical degeneration could potentially be a primary neurodegenerative event that might even drive striatal degeneration. This cell non-autonomous mode of striatal degeneration posits that the loss of cortical innervation into the striatum actually starves medium spiny neurons of critical pro-survival signaling factors like BDNF. And supporting this theory is the even more pronounced degeneration of the subcortical white matter tracts. The white matter tracts, which are the axons of these surviving pyramidal neurons, are almost completely absent in late-stage HD. It's also possible, though, that cortical degeneration is simply a downstream result of striatal degeneration owing to increased glutamatergic activation and excitotoxicity, which we're going to talk about in a second. Another pathological feature that you can't really see here is increased density of astrocytes and microglia, a process called gliosis. Astrocytes are about 70% more dense in the HD neocortex, and part of this increased density is due to the shrinkage of the neuropil and the loss of neurons. You can see that it's almost as if the gray matter has collapsed into the white matter tracts, and many of these cortical layers have been kind of smushed together. Supporting the idea of axon degeneration within subcortical white matter tracts is this recent study by Philippos et al., published in the Journal of PLOS One. So I'm not going to go into detail, but what they found is that the presymptomatic HD patients, so the prodromal stage of HD, So people who have the CAG expansion mutation but have yet to show symptoms, they actually show a consistent trend towards white matter degeneration. So FA, AD, and RD, these are just various measurements that the researchers obtained through diffusion tensor imaging. DTI, diffuse tensor imaging, is basically a technique that measures water diffusion in vivo in the brain of an HD patient, and by measuring the diffusion of water, they can deduce white matter volume. Because normally when water is diffusing, it'll run into an axon and stop diffusing. It'll run into a membrane and stop diffusing. So if there's increased diffusion, that's telling them that there's less white matter, less intact white matter. And basically the summary here is that there's a reduction in white matter density that precedes symptoms and may even precede striatal degeneration. But generally, it's believed that the first stages of degeneration are striatal atrophy and white matter degeneration. The authors also looked at iron levels, which is pretty interesting. The increased iron density in the prodromal or pre-HD patients was speculated to be caused by increased remyelination of damaged axons. This is a process that requires iron. And increased striatal iron accumulation is actually another hallmark of HD, which we'll touch on briefly near the end of the presentation. This is an MRI from a prodromal HD patient. So clearly, even before symptoms have begun, there is whole brain shrinkage with markedly degenerated striatums and also reduced white matter and gray matter densities. So this prodromal HD patient has a smaller brain volume, larger ventricles, dichotic putamen is shrunken, the neocortex is shrunken. It's definitely pretty clear that something is wrong long before symptoms are showing up. Another macroscopic feature of the HD brain has been identified through the delivery of radioactive tracers following by PET. So in this technique, a radioactive drug that binds to a specific receptor is injected into the patient, and the resulting radioactivity is measured in a PET scan, a PET scan. So using this technique, various alterations in gene expression have been found far before symptoms ever appear. So HD patients show, or prodromal HD patients show very early changes in phosphodiesterase A10, PDE10A. So they show these very distinct changes in phosphodiesterase 10A expression up to 25 to even 40 years before disease onset. PDE10A is highly expressed in medium spiny neurons, and this protein functions broadly to regulate cyclic AMP and cyclic GMP. So if you remember, phosphodiesterase just cleaves up cyclic AMP to regulate the second messenger and will basically downregulate it. In HD patients, PET scans show a roughly 30 to 50% decrease in PDE expression 30 years before disease onset. It's very fascinating. Roughly 18 years before the age of onset, there are abnormalities in CB1 receptor density that have been observed. So CB1R, or the cannabinoid receptor, is highly expressed in medium spiny neurons, and CB1Rs function to dampen glutamatergic cortical inputs. So in HD patients, CB1Rs are vastly downregulated. D2R, dopamine receptors, are downregulated roughly 15 years from age of onset, as well as adenosine receptors, which isn't shown here. Also not shown here is glucose metabolism is downregulated. So striatal cells are said to be slightly hypometabolic prior to their widespread degeneration. And in terms of numbers, striatal cells are consuming about 3 or 4% less glucose than control subjects, which that's not hugely significant, but it's still an interesting phenomenon. And finally, and probably the nail in the coffin for striatal cells, is widespread microglial activation, which can also be detected. It can be detected in vivo using these PET scans. And microglial activation is not a good sign. That means these microglia are probably phagocytosing dead or dying cells. And so that tells us at this point, even 10 years before symptoms are arriving, that neurons are dying, and the patient's probably losing striatal cells at an accelerated rate. So before moving into the cellular pathology of HD, we need to just think about and look at the Huntington protein. So the Huntington protein is it's an essential protein that when knocked out, when Huntington is knocked out completely, it causes a severe neurodevelopmental disease that looks a lot like Huntington's disease. It's known to function in many different processes within the cell, but the exact cytotoxic activity of Huntington has actually not been pinned down. There's definitely no shortage of theories. However, there are probably hundreds of theories and ideas put forth that claim to have found the toxicity of Huntington, but no one knows for sure what it is. Anyways, let's look at what we do know about Huntington. First of all, it stains diffusely throughout the cell. It's primarily in the cytoplasm. In HeLa cells, right over here on the left, in HeLa cells, it forms punctate-like staining patterns. In neurons, we see right here in the middle, in neurons, it's more or less throughout the entire cell, and diffuse staining. We also see it right here. This is another stain of normal Huntington staining diffusely throughout the cell. The mutant Huntington protein with an expanded CAG repeat, as we're going to see in the next couple of slides, tends to aggregate into large clumps. We see some of that process happening right here. So what do we know about medium spiny neurons? Medium spiny neurons, abbreviated MSN. MSNs, they are the most vulnerable neuronal population. MSNs are also, of course, the most abundant cell in the striatum. They make up 95% of the neuronal population of the striatum. We know they are GABAergic neurons, so they are inhibitory. There are two main populations of MSNs, those expressing the D1 dopamine receptor and those expressing the D2 dopamine receptor. And this distinction determines where the MSN projects to. MSNs expressing the D1 dopamine receptor project inhibitory efferents to the substantia nigra, and MSNs expressing the D2 dopamine receptor project inhibitory efferents to the globus pallidus. Unfortunately, though, I'm not actually going to delve into this circuitry at all. It has been implicated in HD, but I'm just going to stick with MSNs for now. So MSNs, medium spiny neurons, they express a protein called DARP32, seen in this stain. The selective striatal expression of DARP32 means researchers can stain and view medium spiny neurons very easily. And a typical immunofluorescence assay looks like this. This is a typical medium spiny neuron stain, and DARP32 is the go-to antibody to find medium spiny neurons. MSNs also have a unique nuclear morphology to them. So they basically have very, very interesting and densely staining heterochromatin. So they use this 2-Pro3, this image from the study. They use 2-Pro3, which is a stain of heterochromatin similar to DAPI. And the pattern of medium spiny neuron nuclear staining is distinctly different from resident microglial cells of the striatum. So the glia are much smaller. This would be a glia, this would be a glia, this would probably be a glia, this would probably be a glia. So the glia are much smaller, and their nuclei stain very brightly with DAPI or other chromatin-dependent nuclear stains. In this top right image, all the neurons have actually been labeled using nu-in. It labels neurons, and you can see that the neurons are much larger and they're much rounder than the glial cells. So anything that's not red is a glial cell, and they tend to be smaller and staining much more brightly than the medium spiny neurons. Okay, so what is unique about medium spiny neurons that might explain their preferential vulnerability? And this is a really critical idea. It's a really important question that researchers have been asking themselves for a very long time. And I'm simply going to mention a couple of intriguing data points. First, medium spiny neurons appear to be uniquely susceptible to mitochondrial insults, such as the 3-nitropropionic acid compound, which is a complex 2 inhibitor. This causes striatal degeneration. Mutations in some of the mitochondrial complex 2 subunits is also capable of causing striatal degeneration, and this occurs in an unrelated disease called Leigh syndrome. So inhibition of the complex 2 mitochondrial subunit can actually cause striatal degeneration, as well as complex 2 inhibitors can cause striatal degeneration, suggesting that they are perhaps susceptible to mitochondrial damage. MSNs also receive an unusual surge of dopamine and glutamate from the substantia nigra and the cortex, respectively. No other neuronal cell type receives this combination of neurotransmitters at this magnitude. So medium spiny neurons may be unusually vulnerable to excitotoxicity, and it's speculated that abnormal wiring and perhaps excessive cortical input could cause toxic calcium flux. It's a process called excitotoxicity, right? Since astrocytes are primarily responsible for removing extracellular glutamate, it's also hypothesized that abnormal gliosis, which is observed in HD patients, could result in abnormal glutamate clearance. So a lot of ideas are kind of floating around excitotoxicity, perhaps due to altered clearance from astrocytes or from unusual excessive combination of glutamate and dopamine, or from mitochondrial damage. Medium spiny neurons also receive 90% of their supply of BDNF, brain-derived neurotrophic factor, from the cortex. As mentioned earlier, the medium spiny neurons are connected to the pyramidal neurons of the cortex, and these projections from the cortex, a lot of these projections are delivering BDNF. And so not surprisingly, the striatal cells, these medium spiny neurons, express high levels of TRKB. TRKB is the receptor for BDNF. And defects of transport or even synaptic communication could potentially jeopardize medium spiny neuron survival. In fact, certain mutations in dynein are known to cause striatal degeneration and medium spiny neuron cell death. Anyways, that's a crash course introduction to medium spiny neurons. Now I'm going to kind of move back into general HD pathology. Okay, so this is what Huntington looks like in vivo. This is Huntington in vivo. Again, it's found in neurons, very low expression, maybe not super low expression, but there's lower expression in glia, and Huntington stains diffusely throughout the cytoplasm, and there's a little bit in the nucleus as well. On the right is an image I pulled from the Human Protein Atlas. Human Protein Atlas. It's a really amazing tool if you haven't used it yet. And this is a Huntington stained section of the cerebral cortex from a normal individual. So again, diffuse staining in neurons, nothing too fascinating. What is fascinating is what happens when the N-terminal CAG repeat of Huntington is expanded. You get these insoluble aggregates that accumulate throughout the entire brain. These aggregates are these dark lumps, and they are even found in the brainstem. This is the cingulate cortex. This is another look at the aggregates in the frontal cortex, labeled using EM48. And another look at Huntington aggregates, this time in our striatum. So note that these aggregates are found in many different brain regions. In fact, aggregates have been found in the brainstem and even in the muscle and liver cells. So it's important to note that just because these cells have funny-looking clumps in them does not necessarily mean they're dying. In fact, the presence and magnitude of aggregation is not predictive of cell death. It might actually be the opposite of that. Some research actually suggests that the larger the aggregates, the better the neuron's chance of survival. So in this way, it's possible that aggregates are cytoprotective. These aggregates might be functioning to sequester toxic Huntington into more or less inert clumps. These are called aggresomes when we're talking about them on an acellular level, a pseudo-organelle of sequestered misfolded proteins. The formation and progression of these Huntington aggregates have been observed extensively in mouse models, not so much in humans. In this popular mouse model that I will focus on, these mice contain a Huntington gene with 150 CAG repeats. In humans, the average CAG repeat length is about 42 to 45. But since mice don't live very long, the CAG repeat needs to be dramatically expanded in order to actually produce toxicity within their short lifetime. Nevertheless, it's a good mouse model, and these images represent the typical progression of Huntington aggregation. So at 5 months, over here, at 5 months, basically we see Huntington appears to kind of aberrantly and very diffusely stain the nucleoplasm. So these are the nuclei, and Huntington appears to be aberrantly localized and accumulating in the nucleus and to some degree in the cytoplasm. By 10 months, insoluble aggregates begin to grow in size. They kind of finally they begin to appear, and they materialize. They form these aggregates. These aggregates kind of they simply grow in size from 12 months to 15 months to 21 months. The nucleus begins to kind of disfigure around 21 to 24 months. We see the shape of the nucleus beginning to look a little bit strange. You see that Huntington's almost moving around the edges of the nucleus. And so the initial sites of aggregate formation are in the nucleus. And this is preceded by the aberrant accumulation of diffusely staining Huntington protein. So normally Huntington is diffusely staining. This figure provides a very nice summary of the neuropathological progression of Huntington's disease in the Q150 mouse model of the disease. So we're looking at a sagittal section at bregma 1.7 and bregma 1.8, roughly where the caudate putamen is most affected. But remember, Huntington's disease affects more or less the entire brain. In this figure, we're looking at just a single section of the brain. So we're seeing the spatio-temporal staining pattern of an antibody directed towards the Huntington aggregates. We're looking at where and when they form. At 5 months of age, mice show diffuse nuclear staining of Huntington aggregates in the striatum. So there's diffuse Huntington aggregates, minimal inclusions in the striatum at 5 months of age. And similar phenomenon occurring in the cortex. By 12 months, the striatum has rapidly progressed to dense, insoluble aggregates. The cortex is only showing minimal inclusions, and instead the cortex mostly still has diffuse Huntington aggregate staining. However, by 24 months, we see that the cortex has also been enveloped by dense nuclear inclusions, and there are many other brain regions also showing this phenotype that are not depicted in this figure. Okay, so now we're really going to zoom in and examine some of the ultrastructural images of Huntington. This TEM, transmission electron microscopy micrograph, is from a 21-month-old mouse with 2 copies of the mutant Huntington gene. Each copy has 150 repeats. Labeled NII is the neuronal intranuclear inclusion. NII is neuronal intranuclear inclusion. It's mostly aggregated Huntington, and also various folding chaperones and probably many other associated proteins. The red arrow is pointing towards an unknown electron-dense body. The paper doesn't explain what it is, but whatever it is, it's not normal. It's abnormal. Outside the nucleus, just outside the nucleus, we have this gigantic vacuole, denoted V for vacuole. This is a huge abnormal vacuole just outside the nucleus. And it contains a couple of degenerated mitochondria, indicated M. These are what look like mitochondria that have degenerated. Another closer look at these NIIs, these neuronal intranuclear inclusions. Huntington has been shown to form these insoluble beta-plated sheets that kind of aggregate into long fibrils that you can't really make out here. And nevertheless, they eventually look like this. These large aggregates. Visually, they aren't too informative, but this is what they look like under a transmission electron microscope in the nucleus. The darker stains around it are the heterochromatin. Zooming out a little bit, or wait, no, this isn't zooming out. This is another look at the inclusion body. In this image, the inclusion body has been labeled by a gold conjugated antibody, a process called immunogold transmission electron microscopy. So the vast majority of Huntington is localized to the aggregate, not surprisingly. But there is still sparse labeling of Huntington around the nucleus and even outside the nucleus as well. So this is the nuclear membrane. Also note these very peculiar invaginations of the nuclear membrane. This is very abnormal. You have these invaginations of the membrane. We're going to see more with this later, but these invaginations, very abnormal. And yeah, wherever you see a dark spot, we have Huntington immunogold. And this is the nucleolus right here. This other dark structure is the nucleolus. Zooming out a little bit, we can see that Huntington immunogold labeling also staining the mitochondria. However, interestingly, it almost appears like Huntington is co-localizing only to degenerated mitochondria, interestingly. As you can see, this mitochondria right here, you can see the cristae forming. This mitochondria is perfectly healthy, at least it appears perfectly healthy, and there's no Huntington in it. This mitochondria is degenerating, in the process of degenerating, and there's also a lot of Huntington staining occurring in this degenerating mitochondria, also right here. And you can kind of see this mitochondria is chock full of Huntington. This mitochondria is chock full of Huntington. And maybe some other dark blobs may also be Huntington. This isn't to say that Huntington necessarily caused the mitochondria to degenerate, but it is an interesting co-localization nevertheless. The relationship between Huntington and mitochondrial dysfunction has definitely been extensively studied. So interest into mitochondrial dysfunction, again, is drawn primarily from the fact that complex 2 inhibitor, 3-nitropropionic acid, causes striatal neuron degeneration, and it causes a phenotype that resembles Huntington's disease. So medium spiny neurons appear to be generally uniquely vulnerable to mitochondrial insults. The mitochondria of late-stage HD patients tend to be hyperfragmented and contain abnormal cristae. But at the same time, not all the mouse models are able to detect changes in mitochondria or mitochondrial morphology until very late in disease. So it's possible that some of the insight we've gained from post-mortem HD patients, their mitochondria may be degenerating because there could be widespread cellular toxicity. So just because mitochondria are degenerated does not necessarily mean that the disease began by targeting the mitochondria. If you see what I'm saying, it may just be a consequence of late stage HD. There's probably a lot going wrong in the brain. So in mouse models have not necessarily pointed at the initial degeneration of mitochondria. It could just be a downstream consequence. In this figure, we are seeing Huntington labeled using the EM48 antibody, conjugated to a gold particle again, in a mouse model of disease. So I believe we're looking at the R62 model, which is different from the Q150. The R62 model is basically the HD mouse model on steroids, which is to say the R62 model is very severe and leads to the accumulation of mini aggregates. Regardless, in this mouse aged 27 months, which is very old for a mouse, aggregates are also found in the axons and the synapses of medium spiny neurons. So this is the active zone right here on the right. We see a nice depiction of the active zone. And I know this because it's an electron dense area, and you can see the juxtaposition to the postsynaptic membrane. And interestingly, the aggregates are not really present in the dendrites. So this is a postsynaptic membrane or postsynapse, and the aggregates tend to be near the synaptic vesicles, near the back, and not near the active zone of the axon, except for maybe these spots. But in the synapse, they tend to be found near the periphery. In fact, I'd be willing to bet that these Huntington aggregates are maybe back around where you might find the microtubules, perhaps. We're going to see in a second that a lot of Huntington actually is found on microtubules. And also note this dark blob. This dark blob is a degenerated mitochondrion. And this is also a mitochondrion that does not look very healthy. It's very rounded in shape. And it's also fairly darkly staining, meaning there's probably some Huntington in there as well. This is more of the same kind of images, but from a different research group. Again, the dark spots are immunogold labeled antibodies towards the Huntington aggregates. And the top left is the neuronal intranuclear inclusion body, the NII, and the three other micrographs are axons that contain aggregates near the presynaptic boutons. And these images are also from the R62 HD mouse model. The authors looked at total Huntington localization and actually found that most of the Huntington immunogold staining to be on microtubules. So the vast number of Huntington, which is these dark black bars, are found on microtubules, which is very fascinating. It supports the idea that Huntington is critical for kinesin and dynein-mediated transport, which is actually a leading theory in HD research. And I also have a presentation on kinesin and dynein motor proteins as well. But again, note that you can see the active zone, you see the synaptic vesicles, so we know this is the axon, this is the dendrite, and again, just as the other research group found, the Huntington aggregates tend to be near the periphery, near the back, perhaps where you might find the microtubules. These are some images from the homozygotic Q150 mouse model. So in panels A and B, we see extensive degeneration of cytoplasmic organelles. So these dark spots are Huntington aggregates. The super dark spots are Huntington aggregates in the nucleus primarily. And these slightly less electron-dense blobs, you see right here and right here, are degenerated organelles. They're probably either lysosomes or mitochondria. If you look closely, you can kind of see the remnants of cristae in some of these dark blobs, hinting that they're mitochondria. And interestingly, they all, or most of them, appear to be surrounded or partially surrounded by these empty vacuoles. The authors of this particular study found that some of the aggregates were found trapped inside of mitochondria, and some aggregates seem to be attached to the outer mitochondrial membrane, which right here, for example, you can see a very dark spot attached to this degenerated mitochondria. It's harder to tell over here on the right because there's so much degeneration occurring, but the idea is that there are these empty vacuoles that are partially surrounding degenerated mitochondria and even some degenerated lysosomes. And some of them are colocalized with Huntington aggregates. In panels C and D, we're actually seeing axon degeneration again in the Q150 mouse model. Note the dissolution of the myelin sheath. It appears decondensed. You can actually almost see, you can see some of the spaces between the myelin, almost as if it was unwrapping. We see more dark blobs, like right here. These are, again, degenerated organelles now in the axoplasm. Panel E is a different image from the R62 mouse model. And strangely, the authors of this paper did not find overt degeneration of the axon in the R62 mouse model. In fact, this particular paper was examining differences between the subcellular pathology of the Q150 mice and the R62 mice. And unfortunately, they found that there's actually a lot of differences between the subcellular pathology of these two mouse models, suggesting that they may not accurately be representing the human condition, which is very unfortunate because getting a good idea of what Huntington is doing in the human brain is extremely hard because we mostly only have post-mortem autopsies to look at. But either way, when the end finally comes for the neuron, it collapses into these very small condensed blobs. It typically begins with the contraction of axons and dendrites. So the neurites kind of come back into the neuron cell body, and then the neuron itself shrinks tremendously until it's almost just a nucleus. You can see the cytoplasm is is barely even present, is essentially basically just a nucleus with a little bit of cytoplasm around it. There's massive nuclear membrane blebbing. So if you look at right here, this is the nuclear membrane. This invagination is just huge. Can't see it so much in these other images, but you can see it very plainly in this image. There are numerous electron-dense spots that are like all these dark spots over here. They are likely lipofuscin granules, which are basically the product of lysosomal digestion of lipids. These electron-dense blobs are full of heavy metals and sugars and are generally thought of as being the product or a flag of unhealthy or dying neurons, as well as many other cells. We also see numerous degenerated mitochondria right through here. These are degenerated mitochondria, you can tell from the cristae. We also see severe membrane blebbing itself. In the R6/2 mouse model, there's a distinctly different degeneration of neurons. They tend to just form a very large dark black blob. They're called dark bodies. And they are just basically they're not undergoing apoptosis. These R6/2 mouse neurons undergo a unique form of cell death that's not something we understand, but they kind of crumble into dark bodies, very electron dense blobs. Here's another paper looking at abnormalities in human pluripotent stem cells induced into medium spiny neurons. So it's a really exciting paradigm. They're basically taking cells directly from HD patients, then turning them into medium spiny neurons in a petri dish, and they're looking for subcellular abnormalities. And they found that HD patient medium spiny neurons show a couple of distinct characteristics. But note that these cells are essentially in isolation. They don't have astrocytes with them. They don't have microglia. They are not in the environment of the brain. So a lot of these differences could be not occurring in the brain, but it's a useful experimental paradigm either way. So I'll quote directly from the paper what they found. Quote, TEM, transmission electron microscopy analysis, revealed larger numbers of lysosomes and autophagosomes, increased evidence of mitophagy, and a significantly enhanced occurrence of nuclear envelope indentations in HD versus wild-type neurons, affecting 85% versus 58% of the respective cell population. So they found increased autophagosomes. Over here on the left, we can see increased autophagosomes. They saw increased mitophagy. This is a beautiful image of mitophagy in action. You can see this little blob beginning to envelop mitochondrion. And they also found irregularities in the mitochondrial envelope. These weird squigglies are indentations in the nuclear envelope and very abnormal. The study also found abnormalities in calcium entry into the cell, although I don't have a slide for that. See, abnormalities in autophagosomes is nothing new to HD. Actually, just in a groundbreaking study published in 2010 by Martinez et al., autophagosomes were found to be actually mostly empty in the mouse model of disease. So looking at a Q111 mouse model, a CAG of 111 repeats. And so what they found is that autophagosomes tend to be mostly empty in the mouse model of disease. Interestingly, this image right here, or this entire figure, is these are actually liver autophagosomes. And in the liver, where Huntington is also present, autophagosomes were found to have this very strange and ghostly appearance. So if you look at most of these autophagosomes in the Q111 mouse model are mostly empty. And this is in autophagosomes taken from the liver. So autophagosomes in the liver of HD mice are empty. It's very peculiar. And of course, this phenotype is also seen in the brain. It's not just in the liver. So Q111 mice now is on the left. The Q111 mouse model also has inexplicably empty autophagosomes. These autophagosomes look, ghostly and empty and very strange compared to over here what they look like normally. Normally, autophagosomes have the remnants of degraded organelles in them, such as mitochondria. And although I didn't grab a slide for it, it also seems to be an abnormality shared between mouse models and the human condition. Although there are many serious consequences that could arise from this dysfunction, one is that damaged mitochondria could be allowed to proliferate. And the accumulation of damaged mitochondria can lead to many problems like increased ROS generation and decreased oxidative phosphorylation capacity. Okay, so now let's zoom out. We're zooming out a little bit. We're going to look at some of the cellular events happening in the striatum again. So on the left is the Q111 mouse model, and on the right is an age-matched control mouse. And so stained in blue is DAPI. This is typical DAPI stain. And stained in red are microglia, one of the hallmarks of disease we discussed earlier. And we can see the widespread activation of microglia. And this phenomenon is nicely recapitulated in animal models. So it's likely that dying or sick neurons do not necessarily have the chance to commit apoptosis, but are rather phagocytosed before that happens. So a lot of researchers want to know, how exactly does a neuron die? Does it go through apoptosis like we might think it does? But it's actually probably more likely that the neurons are being phagocytosed by resident macrophages when they become significantly unhealthy. And this is actually probably a good thing, because if neurons were dying and not being degraded, they would be creating a very toxic immunoinflammatory environment for other neurons. So the phagocytosis of dying neurons is probably a good thing, probably. Oops. The appearance of reactive microglia is also a phenotype found in the HD human brain as well. So in the middle panels, or in panels A and C right here, A, B, wait, I don't have C. Well, in these middle panels, A and B over here on the left are age-matched controls. I believe. Oh, sorry. I got lost. Okay, so A and C, so A, C, that's why I was talking about A and C. These two panels are from age-matched controls. Let me turn on my pointer. These two panels are from age-matched controls, and B and D over here on the right is from an HD patient, grade 3 HD patient. And these dark blobs are activated microglia. And we see they're activated. They are what's called reactive at this point, and there's vastly increased numbers of reactive microglia. I believe A and B are from HD patient, and D and E are from age-matched controls over here. Zooming in a little bit, we see that microglia are kind of probing and interacting with neurons, or even actively phagocytosing a neuron, as we see in panel B. You see this microglial processy kind of interacting and feeling the neuron, probably probing it for receptors and seeing if it's unhealthy or if it needs to be phagocytosed. This likely represents the primary mode of neuron disposal in HD. Also of note, these reactive microglial cells found in HD brains are chock full of iron. So I mentioned earlier that HD brains show increased iron in their basal ganglia. Well, when examined post-mortem, as in this image over here on the right, on the left so on the right here, these are reactive microglia in an HD patient. On the left is an age-matched control. You see a similar pattern to what we saw on the last slide. On the right is, we see that a lot of the iron, which is stained in green right here, is actually found inside of the reactive microglia. A lot of the excess iron is found sequestered in these microglial cells. And we see this enhanced iron accumulation in the microglia because it's believed that these microglia are consuming dying neurons, and all these dying neurons kind of have accumulated iron in them, and so the iron just tends to build up in the microglia themselves. So one of the leftover products of a dead neuron is its iron that it had, and perhaps the microglia aren't disposing of the iron as quickly, and the iron actually tends to accumulate in the microglia from all the dead neurons that they consume. And okay, so for my last slide, I wanted to return to the pyramidal neurons of the neocortex. Remember, the neocortex degenerates just like the striatum, but it's not as pronounced. In this figure on the left is a Golgi impregnated pyramidal neuron from a grade 0 HD patient. Grade 0 HD patient, we don't see any overt abnormalities this early in disease. There's normal dendritic spine density, and overall neuron morphology looks pretty standard. However, these second two figures over here on the right, we see a grade 2 HD individual. Pyramidal neurons are in advanced degenerative stages of the disease. These neurons actually show about 30% more dendritic arborization and about 30% more total dendritic area than controls. They also show about 30% decreased dendritic spine density. And perhaps most interestingly, they show 6 times more dendritic varicosities than controls. A varicosity is an abnormal bulge. So they basically have about 6 times as many abnormal bulges in their dendrites. So to summarize, the dendrites of neocortical neurons in HD patients are increased in density. Their spines are downregulated, they're spineless, and they show abnormal bulges that are probably aggregates forming along the axon potentially. So anyways, that's all I have for HD pathology. If you guys have any questions or want me to clarify something or have a request for a future video, please let me know. And thanks for watching. I hope you enjoyed. See you next time.